Thanks, Frank, and thanks everyone for joining in, and thanks to the LBA and Jennifer Brown and Joe San Roma for working with us to host this virtual event. I was having my own technical difficulties, so we appreciate you all sort of hanging in there with us and working with us on this. Now, obviously, we would love to be actually out seeing bees together, but of course, we needed to hold this virtually, but it does provide us an exciting opportunity to sort of expand the audience, people that wouldn't normally be able to travel to our facility to actually share some of the exciting research and progress that has been going on at the lab. So my goal today is to really kind of give a broad update of the things that have been going on at the lab and kind of setting up some of the later talks in the later in the day. So last year at this time, we were fully staffed with 8 scientists or researchers, and now a fully staffed lab, and we've been really busy, even in spite of the pandemic conditions. We had 3 new scientists start in the last year. We also had one big change, and that was what Jennifer Brown mentioned earlier, that Bob Danka retired in July. Of course, we're sad to see him go, but he's very busy and happy in his retirement and still involved in some continuing projects at the lab. However, we've still got this really excellent and really probably one of the most diverse groups that certainly we've had at the Baton Rouge lab, really incorporating all of these different expertise and invigorating some new ideas and really some new targets of selection and whole different avenues of research that we're really excited about. So one thing just to introduce everybody to the researchers at the lab. So we have Ariane Avalos, who you'll hear from later today, Lainey Bilodeau, Lily de Guzman, Kate Eiley, Vincent Rosigliano, and Frank Rinkovich. And you'll hear from both Kate and Frank later today about more specific research. And then I'm currently the acting research leader at the lab. Now, it's important to give some sort of general context, I think, for the day and the talks about our research, and really that comes from the mission of the USDA Agricultural Research Service, which was really our mission at the lab too, and that's to develop, translate, and transfer solutions for agricultural problems of high national priority. And certainly honeybee health and the pollination services that they provide are really key aspects of this sort of national priority. And the problems, including annual mortality rates that commercial beekeepers and hobbyist beekeepers are constantly fighting, is one of these problems that we're really working on developing new and different kinds of solutions. And these are long game, and some have short-term goals, but really the long-term goal is to transfer these solutions that we can develop to help solve these problems and increase the sustainability of beekeeping and have healthier and more productive bees. And the research that we do tends to be on sort of these 5-year plans that we work and develop. And we've just started this past year, the first year of this new 5-year plan with all of these new scientists. And really this, again, is this large, really integrative plan that kind of touches on sort of 4 main areas of research. And the first one is biotic threats. So these are really pathogens, parasites, viruses, and pests. And so there's a lot of research focused on this. Obviously, the main focus has been and continues to be Varroa. And so part of that's understanding individual and social or colony level immune responses. And one of those things includes the Varroa sensitive hygiene trait that we've talked a lot about. And that's still a very important aspect of our research. But we also have research going on viral resistance, development of RNAi tools as potential treatments for some of these threats, in addition to just the biology of some of these resistant traits and the pests and parasites themselves, again, focusing on mites and Varroa, but not just Varroa. There are other species of mites that we have scientists studying. There's also nutrition. That's kind of a newer line of research that we've been working on recently in lab. And Dr. Eiley is going to be talking about these first 2 points, so efficiency of nutrient conversion, and then storage proteins as breeding targets. Then there's another line of research that's been developed on microalgal diets as potential feeding supplements, and that's something that I'm going to talk about a little bit more coming up. Then obviously genetics and breeding, those are two of the first 2 words in our title after honeybees. So clearly that's a major focus of our unit and very important to our mission. But there's some sort of strong genetics work that's been happening, really trying to identify sort of the diversity that exists in stocks and within stocks to really improve our ability to perform selection for specific traits. And sort of coordinating with this is looking at sort of inbreeding effects and specific areas of the honeybee genome that might influence inbreeding that really can inform breeding populations so that we can prevent inbreeding from even happening, but really understand when it's more likely to occur. And then lastly, there's several lines of research on pesticides, including the genetic basis of honeybee responses to pesticide work that's coming up, in addition to sublethal effects of fungicides. And you'll hear more about Frank's work on amitraz resistance in Varroa. But again, really sort of all of this to me really fits into how can we promote bee health as part of sort of this integrated pest management paradigm. And so you might have heard of IPM or integrated pest management before, and a lot of that tends to, people at least think that relates to how you apply treatments and different kinds of treatments. But really, the honeybee should be considered part of that integrated pest management program. And really kind of understanding how bees defend themselves and promoting these traits, I think, is critical to maintaining healthy populations. And it's sort of this key part that we work a lot with here to try and understand these traits of resistance and really promote them and develop new traits that might be of interest. And really then developing new management techniques and strategies can complement honeybee health and natural defenses. So really kind of integrating sort of the bee and bee traits as part of this sort of overall view of how we can work with honeybees to be more productive and healthy on their own. To give you a little bit of an idea of sort of the breadth of research I'm talking about, I just wanted to show a few recent publications that have gone out this year. So even in these pandemic conditions, we had 17 papers published in the last year and many more in review or revision that haven't been published yet. So I'm anticipating even more actually in 2021. But one that just came out earlier this month was this large-scale multi-year effort that ultimately was seen through to completion by Perot Salau, who was a postdoc of mine. And this is really this first sort of large-scale whole genome sequencing effort of different honeybee stocks. So kind of this first step in a long road of research to really identify some genetic differences within and among honeybee stocks so that we can really have a better information of sort of the genetics of the bees in the US. And Frank, earlier in the year, he published an article on amitraz resistance that was detected in populations of Varroa, and he's going to talk more about this line of work coming up. You know, on another mite, Lilia led a study looking at sort of historical trends in this other mite species, these tracheal mites, that provide some nice historical context. Then on the pest sort of side of things, you know, there are more other genomic research looking at small hive beetle and potential identification of target sites that might be useful to develop new insecticides. And then sort of totally shifting gears, Vincent led this study and this work on spirulina, a microalgal species, and looking at how that might serve honeybees in terms of a food supplement. So there's a pretty good breadth here, and this again is just a little snapshot of the papers that were published. There's also several awards and grants that we received in 2020, in addition to the grants that we already have with University of Minnesota and Missouri and a couple other Project 8% grants that just finished up this year. So these are just ones that were awarded in 2020. Frank Rinkovich and Ariane Avalos got Project 8% funding for to really boost and add on this genetic component to this Amitraz resistance project. So that's something that worked a lot with beekeepers during these current sort of our maximized telework stance that we have now to really widely sample beekeeping operations for potential Amitraz resistance and then look at the genetics tied to that. So that's pretty interesting that you know, we're really looking forward to completing. Then the next award I wanted to mention was a team led by Vincent Rosigliano, where the USDA had this ARSX competition, and this is really to reward sort of innovative research. And this team was one of 3 of the winners for this competition, and we're particularly excited about that. And what this research is it's really studying sort of specialized lab-grown microalgae, and in this case it's spirulina here. So we have lab-grown microalgae that we grow in these tanks, and then we're really testing it as a food supplement, both just in terms of general nutrition, but how it might also protect bees against parasites and pathogens. So this is really up and coming research that's going to be continuing for the next many years. And really this idea of microalgae sort of being a potential promising nutritional supplement for honeybees kind of comes from this idea that pollen, you know, pollen is the protein source for honeybees that includes all kinds of nutrients, essential amino acids, lipids, vitamins, minerals, among others. And microalgae seems to have a similar profile to pollen. So the microalgae can actually be a better food supplement when in times of pollen dearth as compared to others potentially. And this is something that we're investigating. And just one little snapshot of research, you know, this is just indicating that as bees eat more microalgae in a cage setting, that they grow more. So they increase their mass. So they are actually, you know, using this food to grow muscle mass. And in the paper that I mentioned earlier, we did find that in many cases, these sort of metrics of bee health and growth match that of pollen. So it really is a promising supplement for honeybees in this case. So Lainey Bilodeau and Ariane Avalos also received an award late this year, and it was an Innovation Fund Award. And what this is, it's a 2-year project, really the goal of which is to modernize the stock identification assay that's currently being used for the Russian honeybee stock identification program, and to update it in terms of numbers of genes or different types of markers that are used, And then it's also sort of using that as a model for how we might be able to look at other genetic or genomic areas of interest, and then eventually lead to potentially other sort of stock identification programs or marker-assisted selection. That's way down the road, but this is sort of where we're thinking, and really this is sort of the first step to get there. So we're really excited about this award, and wanted to share that with you as well. So then lastly, we got funding this year that already went through on oxidative stress and viral infection, which importantly, sort of the real end goal is to develop or investigate this potential antiviral therapeutic, and that's a project with that I've worked on with, led by Daniel Swale at LSU. And then one other award that I received earlier in the year was this Early Career Scientist of the Year award for the Southeast area. And I was really honored to get this award, but to me, it really speaks of the collaborative nature at the lab, and really speaks well to how we all work well at the lab. These kind of awards aren't really just for one individual person, but it really speaks to how well the lab's working together and the productive nature of our lab. So we're really excited about all of these things, and how we're moving forward and progressing. And I just wanted to sort of end with this idea that hopefully translates to into the rest of the talks, and that really that you know, bees are resilient, but we're really working to help make them stronger, and that knowledge of current stocks and new traits is really key for stock development and stock progression. But also understanding more about pathogens and parasites is needed to inform treatments and management and breeding decisions. And so that, combined with the development of new management techniques, can really assist in these natural resistance mechanisms, but also how we manage these parasites and pathogens. And so really that's why our research and our approach is really so integrative and including all of these different facets. So with that, I'd like to thank you again for joining us, and I'm happy to take any questions and comments that you ever have. Thanks again.